Hi, everybody. Welcome to the second research Q&A drop-in sessions, where we really encourage the community to join us every month and just have a chat with Armel and understand research, get to know what we're up to as a foundation, and just ask questions generally about Myhre syndrome, because genetics, as we all know, is complicated. And it's a great time now to ask the expert, and Armel will be able to guide us through any questions that we have. So I'll hand over to Armel, but thank you all for joining us. We really appreciate your attendance, and this will obviously be recorded for people to catch up on next time as well. So Armel, over to you.
Hello, everyone. Welcome for today. I think, Kate, you need to allow me to share my screen.
There we go. Get the basics right.
Yeah. So I'm just going to repeat that it's really something that I want to be very organic and have you ask your questions, interrupt me, tell me, no, I don't understand that, or why do you do that, and please go back, or ask any questions. There are no stupid questions, and just go ahead. And it's, as Kate said, it's more a conversation than it's not a lesson. We are not in an auditorium. It's really, I want you to feed me with what you want to know, and I need to know your level. So today might be a bit impressive sometime. I'm going to go really like a crash course on very basic science. And if you are not interested, just tell me, no, we want to know something else. Or if you have questions, just interrupt me. So I'm going to share my screen. Of my screen, do you see the presentation?
Yes.
Okay.
Yep.
Okay, so I'm going to go in slideshow. Okay, so just as a starter, I just want to remind you, well, the strategy at the foundation is based really to work in parallel on 4 different aspects that I call the pillars. So the preclinical toolbox is everything we need to study and develop treatments before going to human. The clinical readiness is all the tools we need to evaluate the efficacy of treatments in a patient, in human. The third pillar we work on is the treatment discovery. So we start immediately treatment discovery. And the fourth pillar is interacting with the regulatory, so the FDA, the EMA, and the industry to really favor and accelerate all the interaction with this partner. Those partners are really essential to develop treatment. So today I'm going to explain things so that you can understand the choice we are making and we will make in the future about treatment discovery. So this third pillar. So what I would like you to understand is that the road to treatments is really studying all the different structure in biology, and starting from the atom, going to molecules, going to the molecules get together to finally make organelles, and the organelles get together to make a cell, then the cell group, different type of cell group to make tissue, then to make an organ system like the cardiovascular or nervous system, and all these systems together make a human. So what you have been receiving a lot of information by the people that study from, let's say, organ maybe to organ system and human. So those are the medical doctor and the clinicians. They work on at that level. Here you have the biologists that work here, and lower, like from the atom to the molecules to the organelles, well, atoms to the molecules, that's the chemists. They are fundamental. And then you have the biochemists that are in between those two molecules and organelles. So I call that on the road to treatments, because a treatment up to now, before the gene therapy and cell therapy, it was based on small molecules, and small molecules are molecules. So the people who make molecules are chemists. So we need to have chemists at the Myhre Syndrome Foundation, I mean, working with the Myhre Syndrome Foundation. On the road to a treatment, everything starts with a chemist. Then the chemist works with the biochemist, they create molecule. Then the biochemist is going to study a little bit more the molecule in relation to the biology. And then this molecule will go in the hands of the biologist. The biologist will check at the level of a cell or at the level of tissue if this molecule that was created by the chemist is working. And then if everything is good, we can give that in the hand of a doctor at the end, of the clinicians, to test it on humans. So that's really, it's a kind of step by step. This molecule that is created by chemists is going to go from one hand to another up to the clinician. But you really need to have experts in all these different steps to be able to go from the beginning to the end. Oops, sorry, I'm gonna use the other one. So I'm going to start in the same way. I'm going to start from the molecule up to the last part. Not everything today, just the first part. So the molecules that are in living organisms associate together to create what we call the macromolecules. And there are 4 types of macromolecules in living organisms that are I'm sure everybody knows that: the proteins, the lipids, the carbohydrates, and the nucleic acids, which is DNA and RNA. So you can hear those words even on TV, or if you go to the sports club, they tell you, did you have enough proteins, or we want to decrease your lipids, and they talk about carbohydrates. So what's that? The macromolecules are association of smaller molecules put together one after another. So the proteins are made of amino acids, the lipids are made of fatty acids, the carbohydrates are made of sugar, and DNA or RNA are made of nucleotides. Well, it's nucleobase combined with sugar. So those 4 types of macromolecules, when they are put together, they are going to start to form organelles. So for example, if you go to a cell, you have the nucleus, which is inside a kind of ball inside the cell. All the DNA is here. The membrane of the cell, which is around, is an association of lipids, proteins, and some sugar on top of it. So those macromolecules get together and start to form organelles. And those organelles all together, I think, Christine, you should put yourself on mute, or Kate, if you could mute Christine. Okay. So all these macromolecules form structures that form organelles that form the cell. Just for you to give you an idea about how it goes up. Now, I'm going today to focus just on not all the macromolecules, but just on the DNA, RNA, and proteins. So I just told you that in a cell, there's the nucleus. In the nucleus, you interrupt me if I go too fast or if there is something you don't understand. In the nucleus is the DNA. The DNA, just before a cell separates in 2 to proliferate, they compact, the DNA compacts, makes really becomes really smaller and forms chromosomes. I'm sure you all heard about chromosomes, so I'm not going to go in that direction. But the DNA that is so inside some forms of chromosomes have different parts, and one specific part is called specific type of DNA, segment DNA, is called a gene. And those genes are in fact the recipe to build proteins. there is a lot of other parts of the DNA that are not made that the purpose is not to make proteins. There are other parts of the DNA that are going to be regulatory. So the DNA is not only genes, but there is a part of it that is dedicated to different genes. So this gene I just said is the recipe, it's the sequence, it's the code, it's the lecture to create a protein. The DNA is made of 4 different nucleotides. I told you before, it's a macromolecule, and the base of the macromolecule is really associating smaller molecules. And the DNA is made of only 4 nucleotides. The adenine, sorry for my French accent, cytosine, I don't know, thymine, and guanine. I think I should say 9 instead of in. So the thing is that the association between the nucleotides are not random. It's really, you always have G that go with C, so guanine with cytosine, and the adenine with the thymine. So it's always, you will not have another type of association. So it's always C, G, A, T all the time. So this sequence of different letters, let's say, it's easier to say, the different nucleotides gives in fact the recipe. The problem is that the DNA is in the nucleus and cannot exit the nucleus. It stays here in the nucleus, and the DNA will stay there for the genes. So what we need, and the proteins are made outside of the nucleus. So we need a kind of messenger that will reproduce this recipe, this sequence in the nucleus, and go outside of the nucleus in the cytosol to create the protein. So this messenger is called an RNA messenger. There's a don't look at that. I'll talk about that another time. So the messenger is created, goes outside the nucleus, and then there is a machinery that reads this sequence and creates the protein. So to create the protein, they are going to read the letters, the nucleotides that were in the sequence, and to each nucleotides are going to be associated an amino acid. So the amino acid is the building block of the protein. And to the nucleotides go by 3. And when you have those 3 letters, it corresponds to a very specific amino acid. So if you change one of the letter, as in for Myhre syndrome, there are some of the letters that are changed, that's the mutation. Then you will change the ball here. You might have, instead of a yellow here, have a red one. And then you have a problem because the protein is not the same. You have a different amino acids. So that's what when you read on the paper that you have, for example, I500E, it means that you had the ball that was called isoleucine here, but because you change one letter here, now it's not an isoleucine anymore, it's a valine. It becomes a valine for some mutation in Myhre syndrome. So you don't have the right ball here, so the protein is going to be different. That's just a small aparté to explain your the mutations, where the consequence of a mutation. But what I want to attract your attention is here. So we have so many different genes that are going to create so many different proteins. Why do we have that? So we have between 22 and 25,000 of genes in a human being, depending on who's counting. And those more than 22,000 genes are going to be able to create more than 400,000 proteins. So you can imagine that every cell is not creating the 400,000 proteins. Everything is going to produce certain proteins and not others. And that's the reason why you have cardiomyocytes, for example, cells in your heart that are not the same as the cells in your brain, not the same as in your gut, because the proteins that are created, so it means the genes that are expressed. You tell me if there are some words that you don't understand. So the protein that are created, that will have a function, that will be part of the membrane, of the shape, of the motility of the cell. All those proteins are different from one cell to another. But now the question is, how come that a cell can have, let's say, protein A, B, and C, and the other one will have the protein X, P, and W? Where does it come from? Well, how the cell decides which proteins are going to be expressed? So it starts from the beginning. It starts, well, there are several levels of regulation, but the first level of regulation is at the DNA level. So when is the gene going to be read, to get messenger RNA? So how do we do that? There is an enzyme that is called the RNA polymerase that's going to go on the DNA and start reading the DNA and create the messenger RNA. But imagine if you have all these RNA polymerase, they can go to any gene and start making mRNA. So you need to regulate this RNA polymerase, sorry. So because otherwise all the genes can be expressed. So to do that, we have other proteins that in gray here is the RNA polymerase, and in blue here is what we call a transcription factor. Because the fact to create this messenger RNA or RNA from the DNA, this process is called transcription. So a transcription factor is going to regulate the transcription. And those are proteins that comes on the DNA around the RNA polymerase, and they are going to either block the polymerase or stimulate the polymerase. They are going to say, no, you cannot transcript this gene, or yes, please transcript the gene so that we have the mRNA so that we can create this protein. So those are the first regulators. Let's say, I simplify, but among the first regulators on which protein is going to be created in the cell. So there are different types of transcription factors. Here you see the DNA, here or here. So you have different types of transcription factor. And I think there is one that you all know. It's the SMADs. So for example, the SMAD4. So the SMAD4 is a transcription factor, and it's going to bind on the DNA and help give a signal to the RNA polymerase, go or not go. The SMAD4 and also it's a whole family, the SMAD3, 2, and 4, they are made with a kind of a ball, a rope, and another ball. So that's what we call the MH1 here. So that's the protein, that's the SMAD4 protein. You have a kind of ball here. That is the part, we call that a domain, that is going to bind to the DNA, like here. So you have one foot on the DNA that's going to communicate with the RNA polymerase. And then you have the kind of rope, a linker, and another ball at the end, which is called the MH2 domain, that's going to interact with other transcription factors like SMAD2, SMAD3, but many others. So if you look here, you would have the SMAD4 that has a foot on the DNA, and the other part, the MH2, interacting with other transcription factors. Is it clear? Did I lose you, or are you okay?
What does everybody think? I was going to do like a really basic summary if I'm understanding it correctly myself.
Yeah.
So like the RNA polymerase, that's like then reading the chapter in the book. That's the right chapter. I've gone here. I want to create heart muscle. It's relevant to cardiac. The regulator is the regulator and the transcription factor. Is the transcription factor the regulator as well or not?
Yes. Yes.
So that is like your counsel, your body counsel saying approve, deny, yes, proceed, do not proceed. Yeah. So it's like reading the chapter book, am I allowed? The transcription factor says yes or no, the regulators agree, and off they go.
Exactly. It's like the foot when you are driving a car. It's the transcription factor is the foot that is going to push the brake or push the accelerator. So it's around, you see the polymerase is here, ready to read, ready to read the DNA. Yeah. So this chapter, as you said, the recipe is here. The polymerase is here, ready to read and create the messenger. And around that are the transcription factors holding the polymerase, saying, okay, you can go, no, you cannot go. I block you, this gene is not going to be transcribed, and the protein is not going to be made. Or the transcription factor says, go, 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 and pushes the polymerase.
Great, okay, that makes sense. Everybody else, is that clear? Everybody, feel free to put in the comments if you don't want to unmute.
Can you repeat that last summary of this picture, this 3D model again, what you just said? I thought it was good, but I was zoning out for a split second and I missed what you said.
Problem. So to if I go back here to the even one more or two more. So here we need, this is the gene, so that's the chapter of the book to create the protein. And since the DNA cannot get out of the nucleus, it stays in the nucleus. Well, let's say, to simplify, usually the DNA stays in the nucleus. So we need to get a messenger, to create a messenger, to go in the cytosol where you have all the machinery to create the protein. We need to get it out of the nucleus. So we are going to create a messenger RNA that will go out and give the message. So to create this messenger, you need to open the DNA, which is usually closed, as you can see here. It's a double helical. So you need to open it, and you need to have a machinery that go on it and read the chapter and create at the same time the messenger. So this reader and creator and the one that builds up the messenger is called the RNA polymerase. It's going to create the polymer of different nucleotide that's going to be the RNA. So it's an RNA polymerase. But what I explain is that if you open all the genes and you start to put RNA polymerase on the genes, all the genes are going to be expressed. So you need to regulate that so that you have, in the neurons, you will have certain proteins. In the muscle, you will have other proteins, because if you look at the neurons and the muscle cell, they are totally different. And they are totally different because the proteins that are inside, that are created inside the cells, are different. So how does the cell understand and knows and regulates which protein is going to be created and which one is absolutely not going to be created? So for that, you need to regulate the polymerase. Otherwise, it will make the transcription of all the genes. And to regulate the polymerase, go or don't go on the reading this chapter. It's what we call the transcription factors. I mean, it's easy to remember because reading the DNA to make an RNA is called transcription because there's a code, so you are transcribing the code. And to regulate that, it's transcription factors. So for once, the name is logical. So you have these transcription factors that binds to the DNA on one hand, binds to other transcription factors. It's not only one transcription factor. They usually work in complexes together, and they bind all together. And by that way, either they block this polymerase here, or they can push it and go ahead. And I was explaining that there are many different transcription factors, and they have different type of transcription factor. And the SMAD, notably the SMAD4, is a type of transcription factor. And there is a kind of big ball. So that's the protein, okay, of SMAD4, which have amino acids on one end that are making helix. So certain type of amino acids, because they interact in a certain way and because of their structure, are going to create, instead of a rope, they are going to turn on themselves and create helix. So that's one end of the SMAD4 is the MH1, and this group of helix is going to bind to the DNA. Then you have a linker, so it's other types of amino acids that are making another structure of the proteins, not helical, but that's going to be beta folded. So it's a kind of more or less straight rope that goes up and down, up and down. And then at the total ends of the protein, you have again amino acids that triggers this helix structure. So the thing is that the SMAD4 is very difficult to study because of this linker that is huge. You can have the SMAD4 that expands or regroup. You can have it can move. The mutations that triggers Myhre syndrome are here in the MH2 domain. And they are rather at the end in the helix number 5, so rather at the end. So if the mutations are here in the MH2 domain, and the MH2 domain function is to interact with other cofactors, other transcription factors or other proteins, it means that in Myhre syndrome, you have an issue with SMAD4 interacting with other proteins. So we met last week a great scientist and her team. Her name is Maria Matias. She's in Spain, she's in Barcelona, and she's studying the structure. She's a chemist, and she's studying the structure of SMAD4 and the implication of the Myhre mutations in the structure of SMAD4. And what she has discovered, that's her work from another part of SMAD4. That's the MH1, but she's going to publish on the MH2 with the Myhre mutation very soon. So what she observed is that the MH2 domain. So I told you the SMAD4, it's one ball, the linker, second ball. The mutation of Myhre syndrome are in the MH2 domain, which is the domain that is going to interact not only, but can interact with SMAD2 and SMAD3. And what she observed, if you go to really the amino acids that are in the domain of SMAD4 and that are changed from one to another, so an isoleucine to a valine, for example, or an arginine to a cysteine, you are going to change really the interactions with the DNA, the interactions with the other proteins. And what she saw is that the mutation triggers a bigger link between SMAD4 and SMAD2 and SMAD3. So this complex here stays longer and is more stable than it should be. So what does that mean? It means so that's that's the general TGF-beta. That's one example of what SMAD4 can do. So SMAD4 can interact with the SMAD2 and SMAD3 and form a complex. And what Maria Macias and her team, that are really very good scientists, they saw that this complex here stays longer together and is more stable than it should be, which means that when it goes into the nucleus on the DNA to interact with the polymerase, it's going to stay longer. So the effect of SMAD4, instead of coming, okay, one polymerase can go, it's going to stay, and the polymerase is going to work way more than it should be. The SMAD4 is also interacting with other cofactors and blocks the polymerase on other genes. There is one study that shows that they remove the SMAD4, they knock out the SMAD4, and what they saw is that when you modulate the SMAD4, you have a difference in genes that should be at a certain level and that are up-regulated. They noticed 595 proteins that are higher than they should be, and more than 800 proteins that are lower than they should be. So if you modify this SMAD4, you are going to act on thousands of proteins. And that's one of the reason why there is this heterogeneity in when you touch SMAD4. You are touching development, so neurodevelopment, cardiac development. You are touching differentiation, so the cells that are in the embryo that are going to be transformed in bones, transformed in neurons, are going to not be transformed the right way. So that's the differentiation. SMAD4 act on cell cycle arrest. So there are some cells that should stop growing and die, and some other that should continue their differentiation. And if you modify SMAD4, those cells that should die will not die, and other that should not die are going to die. So that's the equilibrium between the cell cycle arrest and the apoptosis. And if you touch this equilibrium, you touch the difference, the development, and you touch the cancer tumorigenicity. And all those genes that are also regulated by SMAD4 act also on the migration. So that's there's a lot. When you touch SMAD4, you touch in so many different things. And we usually talk about, yeah. Is there a question? No.
No, sorry. Airplane.
So when we talk about SMAD4, we always hear about TGF-beta signal is BMP signaling, but there is another one that is very strong, which is the Wnt signaling, Wnt frizzled. And their SMAD4 interact not with the SMAD2 and 3, but with LEF and beta-catenin. And everything, so the SMAD4 is going to interact with the SMAD2, 3, with the LEF, with so many other, with the notch, with the, even with RNA, but I'm not going to talk about that today. And there is a very fine equilibrium between all those different pathways and all those different proteins that are either created or blocked the creation in the cell. So the level of SMAD4 is extremely important in a cell. And if you touch it just a little bit, then you create an imbalance. And that's when you trigger different type of diseases. So the SMAD4, I hope you understood that it's involved in the creation of so many different proteins, or in the blockage of the creation of so many different proteins. It's regulated through different signals, and everything is a very subtle balance. If you touch the SMAD4, you are going to modulate the immune response, the T cells activity, inflammation. Now we know that SMAD4 is involved in IBD, for example. You are going to trigger a lot of tumor formation, because usually SMAD4 makes the creation of proteins that are going to prevent the tumor formation. So if you remove SMAD4 or decrease SMAD4 a little bit, in fact, it's like there is this SMAD4 on the brake of your car all the time, and suddenly if you just pull a little bit your foot from the brake, then your car is going to go faster. So that's the problem. If you start to remove a little bit SMAD4, the car is going to be driving faster towards tumors. So it's really very frequent in colorectal cancer, pancreatic cancer, gastric, breast, and other type of cancer. There are congenital heart disease if you have a loss of function, or if you have a gain of function, like in Myhre syndrome, for example. You have arterial and venous malformation. You have neurodevelopment and psychiatric consequences. And there is a very nice study on microglia, which are cells in the brain, showing that SMAD4 is essential for microglia to work properly. And if you disturb the microglia, you will have neurodevelopment issues and even psychiatric issues. You all know that SMAD4 is involved in fibrosis. If you remove SMAD, you know that you have 2 chromosomes in every cell, one from your dad, one from your mom. So if there is one of the 2 chromosomes that have a mutation, only one of the 2, which is called autosomal dominant. So if you have, like in Myhre syndrome, in some disease, we just say, well, we are going to remove the one that has the mutation and let the other one just work, and that will be enough. In the case of SMAD4, if you remove one of the 2 SMAD4 from one chromosome and not the other, this is not okay. The SMAD4 is not going to work properly, and it triggers the juvenile polyposis and cancer and other things. This phenomenon is called haplosufficiency or haploinsufficiency, which means that sometimes for some proteins like the SMAD4, you need to have both to be able to function. If you remove one, then you go into disease. It's also involved in adipogenesis, angiogenesis, cartilage growth, muscles. I didn't have enough space to write everything, but you can imagine is if you have more than 500 proteins that are upregulated and 800 that are downregulated when you modify the SMAD4, you can imagine all the consequences. So knowing all that is really what we need to pay attention to choose the strategy and the type of treatment we want to push. For example, pushing towards ASOs that will remove one of the SMAD4 that is mutated and leave only the other chromosome, we know that's not a good idea, because then you are going to trigger juvenile polyposis or trigger other things. So really understanding how it works, how the SMAD4 works. I'm going to stop sharing just for a moment to have your face. Really understanding how it works, the different implications, the structure of the protein, the chemistry, really replacing an isoleucine by a valine or an arginine by a cysteine, and looking at the structure as different teams, and notably the team from Maria Macias, really looking at the interaction. That's a key knowledge that we need to have from the beginning, so that we are not going to push a treatment that has no reason to be there and that is going to be deleterious for the people. So that's the reason why at the beginning I explained you that we need the whole process. We need chemists, we need biochemists, we need biologists, and at the end, to put in their hands the right treatment, we need the clinicians. But the beginning is really fundamental. And the understanding the vast action and function of SMAD4 is really a good way also for us to choose between the different strategy and treatments, so that we don't go in something that is going to be toxic for the patients. I have other things, but I talked already too much. So is there any question? Is there something you don't understand, or something you want to know more that I didn't talk about just today? I know it's a lot. You need to digest just for 5 minutes, maybe.
I've got a question while people are maybe thinking about theirs. So, you know, you said that over stability, it stays longer, the interaction with that SMAD4, 2 and 3 that Dr. Myhre had identified. In terms of how that presents itself on that range of symptoms we see across different patients, is it that there's a difference in the level of that stability or over stability, or is it that it's the interaction with all of those other things you said? So is the variability in that stability? If that makes sense.
Yes. So what she showed is that depending on the mutation, the stability is going to be different. She compared the I500V to the R496C, and she saw that the strengths and the time that the complex stays together is different from one mutation to another, and totally different from the healthy SMAD4. So this really the fact that you change one of the one of the amino acids is going to change totally the interaction between the different atoms, and every single mutation is going to trigger a different ball and then a different interaction between the atoms. So it will be slightly different. What we see is that globally it's stabilized, but it stabilizes in different ways. And she just studied the interaction of SMAD4 with SMAD2 and 3, but there's also all the other interaction of SMAD4 with LEF, with all the other type of transcription factors. But if we can really identify for that mutation, it's that amino acid that is different. So they have kind of how can I explain, computer, if you want, that makes all that recreates all the different interactions in between the amino acids. It's different forces that brings the molecule together or push the molecule, and it depends on the, well, I'm going to go deep. It depends on the electric charge of the amino acids. You know that when you have, it's like magnets, if you want. If you have magnets, they can attract each other, or if you reverse them, they can push each other. So when you change one of the amino acids, one of this little ball, you can create another force, either attract more or push more, which means that the structure is going to be slightly different first, and the interaction with another protein is going to be also different.
Okay.
And that's really where we need to understand better, so that we can project and know exactly that molecule is going to be okay, that molecule will never enter there. So that's it's a good way. It's what we call in silico. So via computer, the computer can recreate this 3D structure, and you put some small molecules, so either drugs that are already on the market, or you create new drugs, and then you can put them in the computer, and the computer is going to see with the force, is it going to be able to interact between those two, SMAD2 and SMAD4, and detach a little bit the interaction. Is it going to increase the interaction? So that's really that's the beginning of everything. Starts with understanding the structure of the protein SMAD4 when you have those mutations.
Okay. Questions from other people? I know it's a lot to take on, as Amel said.
There was something in the chats. Oh, was it?
Oh, I think that was just
Oh, no.
yeah.
That's something else.
And I guess following on from that, without getting even more complicated, is that where you see a big difference with the same mutation and a huge difference in symptoms, is that then more to do with the interaction with the other cell, other things going on?
Yeah, totally. That's everything starts from there.
Yeah.
So if you change, if you have the mutation even at the same spot, the 500, if you change for an amino acid valine, or if you change for an amino acid methionine or a threonine, all the different amino acid valine, methionine, threonine, they have a different structure. So they have a different way to push or attract the other parts of the molecule or the other molecules. So every time you change for an M, for a T, for a R, for a V, you are going to modify this structure. And knowing that explains you exactly what's happening with your mutation. You can see it with your eyes. You can see that's instead of having the protein like that, you change one valine and then the protein is, no, I shouldn't do that. And then the protein is going to be slightly different, more open, more closed. So it's really a visualization of the consequence of changing one amino acid to another, even at the same spot. If you change one that is going to be extremely charged positively, it will change the molecule, the SMAD4, in a different way than if you change it for something, an amino acid that is charged negatively. And then the interaction between the SMAD4 and all the other proteins that you can have is going to be modified, because if you have something that becomes suddenly more positive, then the interaction with something that is positive is not going to be possible. So that's really, it's not the position. Well, the position is important, like the 500, the 496, that's important. But what is also very important is the change in this little ball, in this amino acid. In what type of amino acids you are going to change.
Why do we see, let's talk a little bit about the progressive nature of Myhre. So given all of that we know that those things start early, they're in us, in ourselves or our babies from the word go. What's the interaction that's happening that means those things can get worse over time? You can see more expression.
So you have an accumulation of stress, of proteins. You can have, there's also the fact that at different age, your cell is supposed to read different genes. So when you are 3 years old, in your brain, the cells are going to create proteins or read genes that are not the same as when you are 10 years old or 15. From after 2 years old, you are supposed to lose some of the structure. When you are born, you have a lot of neurons, and you are going to create more and more and more and more up to a certain age, and then you are going to remove some of these neurons and to facilitate others. And in autism, for example, there's a large part of those neurons that should die, that do not die, stays, so that you have some other neurons that should be regulated or going down, that are still stimulated by all the others. Because with the age, it should have read some genes that they didn't read, or that some genes that they read too much and they shouldn't have read those genes anymore. So really, depending on the age, you are going to express certain proteins or not, so that the development, you evolve all your during all your life, you evolve. I mean, I'm not the same as when I was 25 years old last year. So that's really very important, is depending on the age, you are going to read some genes and other not. And notably for autism, the fact that they do not kill all these neurons, they can be, for example, overwhelmed by too much sensory stimulation, because they get too much, because all the neurons are still there and the inhibitory neurons are not enough. So they cannot compensate an overstimulation of noise, of whatever the stimulation they are sensitive to.
Okay.
And is it the transcription factors that aren't working because they're supposed to regulate, but they're not, right?
Yeah. A gene is expressed because a transcription factor says to the polymerase, you can go. And because the DNA is open, also there is another regulation, level of regulation. But what tells the polymerase make the transcription into the messenger RNA, or no, don't make the transcription, that's the transcription factors that tells the polymerase what to do. And SMAD4 is there. He's the one going to tell the polymerase, okay, you can go on that gene and do it, or no, you cannot start on that gene, stop. So if you remove this regulator, then the cell starts to either express too much or don't express.
Is it known what the transcription factor is in autism?
Oh, there are hundreds of them.
Oh, hundreds that don't work right.
Yeah. Hundreds of different genes that are involved. But SMAD4 is one of them.
Oh, that was my next question. So okay, so there is the association in the SMAD4 and the autism regulation problems.
Yeah, through different other genes. So that's something I really want to push also, is to go convince the teams that are working on the different genes of autism to include SMAD4 in their study for autism, but for learning disability and neurodevelopment. There are some good teams working on that, and I'm sure we can include SMAD4 in that also.
Great.
They do.
Feel free to ask any questions about other things we've not covered today.
Yeah, because I was supposed to talk about biomarkers, in fact.
No, we know it's like this is what's great about these is like you just you can have a little bit of knowledge, but you dip your toe and then it's so much to understand. And I know for sure I'm going to be revisiting this recording and just understanding
because I was supposed to talk about, do you see that?
Yeah.
So I was supposed to talk about the biomarker on how to measure the disease progression and explain the different molecules that are in the blood and how we can measure them and follow the progression of the disease through the level of those molecules that we take in the blood. And I wanted to insist on the fact that it's very important that if you go to the conference, that you participate in the combined brain collection, blood collection. That's and we need to compare the blood of your kids with or of yourself if you are a patient to siblings, to people that have rather similar molecules in their bloods, except for the difference would be because of SMAD4. So that's that was something I wanted to talk about. And now the biomarkers, people start from the blood from the patients and identify the markers, and then try to identify if we can find the same in the mouse model. 10 years ago or 15 years ago, they used to start from the mouse look at biomarker in the mouse and then go back to human. But now we know that there is such a difference between the mouse and the human that usually the biomarkers that they were identifying in mouse didn't work in human because we are not mice. But it took 10 or 15 years to finally understand that we need to start all the studies from the human, from the patient, and not from the mice.
And Amel, we have our next session next month will be a week before the conference. So we can perhaps touch on that a bit more for people who are going to the conference, talk about the biomarkers in a bit more detail then. It will still be very relevant as well. And more information will be coming out tomorrow on the opportunities at the conference for collection as well. Any other questions about anything, not just related to all the complexity?
I had a question about Maria Macias. What kind of researcher is there? What level is she considered? Like a biochemist or you know?
She's a chemist. She's a chemist, and she works with biochemists and biotechnology people. But she has collaboration with many other biologist labs in New York, in China, in Germany, and she's everywhere. She's the big lady of SMAD4 and the SMAD in general, but more the SMAD4. She's a big name, and she's great, and she's a very nice person. In addition to being let's say the queen of the SMAD4 chemistry.
And in a great city, Barcelona.
And Barcelona, yeah.
I don't know, maybe we should have a conference there.
Yeah.
Okay. Date? Did you hear that?
2026. We'll start working on it now.
Yeah, I think that would be a good idea to do the first scientific Myhre syndrome scientific conference in Barcelona.
Well, yeah, scientific and patient. Like, let's just take over a whole week. Oh, yeah.
Yeah, we can put them one after another, the patients and then the scientific conference. So there is an interaction between the scientists and the patients. It's very nice.
Yeah. Okay, I'll start looking up.
So I think I really just knocked you out with this this explanation.
You did, but it was great. I mean, it was really good. It's a lot, but it is very good.
So the other that you don't have any other question, or is there anything that you want me to talk about next time, that you have questions about something maybe totally different? No, you understand everything now. Well, I'm going to repeat that over and over and over, not in the same way, in other ways, because I just presented you one part of SMAD4. SMAD4 is way more than that. There are also other factors, other interactions. It's a great protein, very complex protein.
Well, I think that's what you're giving us the window into, is that the level of complexity and interaction that is more of a challenge. You know, we're so fortunate in many ways of our small number of mutations, but we are on a complex gene. So
and there's many levels of regulation of SMAD4, because I just explained that SMAD4 regulates other genes, but SMAD4 itself is regulated by others. So it's a kind of mix of loop of and it's complex, and we need to be very careful understanding everything, so that when we decide to touch something, we know that it's not going to trigger an imbalance in all these other loops. That's the reason why in oncology, or when you touch to transcription factor with small molecules, you have so much toxicity, because you start to to trigger imbalance in all these very subtle expressions, and then you go in cytotoxicity. And for, if you want to treat a tumor or a cancer, it's maybe for 6 months or a year, so you are more or less okay with this toxicity. But when you have kids that are, or patients that are going to take this medication all their life, then you cannot go into that toxicity anymore. You cannot be under chemotherapy for 15 years or 20 years. That's not, or all your life. That's not possible. So we need, in chronic disease, we need to be very careful with all the rest, not touching all the balance. So that's the reason why the first priority was to do gene editing, because just correcting this error, you don't touch at the level of SMAD4. You just correct that the structure of the protein is going to be okay, but you don't touch at all the other things, all the amount of SMAD4 that you have, you don't touch that. So you respect this balance. You just correct, instead of having the wrong small amino acid, you put the right one. That's it. So you don't interfere with anything else.
Yeah.
So gene editing is changing the nucleotides or the amino acid?
Yeah, exactly. The nucleotide. So since you change the nucleotide, then the messenger RNA is going to have the right code, and then it will trigger the creation of the building up the right protein with the right amino acid.
We have that technology already that exists to do that? To change.
Yeah, the technology exists, and we just we did an RFP, so a request for a proposal, and we have different teams that are going to send the proposal to us, and we are going to choose one team to do that.
And we are expecting there wouldn't be toxicities associated with that, I guess.
Yeah, there's always toxicity with everything, but at least it will be toxicity due to the technology and not the technology plus the equilibrium, modified equilibrium of SMAD4. For example, if you use ASOs, so that you are going to remove the messenger RNA that has the mutation, there is a lot of toxicity due to the technology itself. But in addition, you would have toxicity because you are going to create an imbalance of the SMAD4 expression. So already, if we have only the toxicity from the technology and not from the fact that you modify the SMAD4 balance, then it's already a better way, because you decrease the total toxicity. And the other thing that we are looking at, it's not correcting the SMAD4, not correcting the gene, but going to the consequences of the disease and trying to act on the fibrosis directly. So that's another. Usually, what you do for the treatment of a disease is either you go at the origin, so the gene, or you go at the messenger RNA level, or you go at the protein level, or you go at the consequences, the process that are modified after the fact that you have a protein that is not functioning. Then you have processes on other cells or in the same cell that are modified. So you can go also for as a target for the consequences, like the fibrosis is one of the consequences. So we try to go at every different levels. Not the messenger RNA, just the DNA, the protein, the protein interaction, just the way it interacts with all the other complexes, and the later consequences. Because the fibrosis, what I believe also is that what you see as the fibrosis in the lungs or in let's say in the body, That's something that is okay to observe, but I think you can make a parallel between the microglia and the astrocytes that are in the brain and the fibrosis. Maybe there are some process in common with the fibrosis that you see in periphery, and something in the brain with the microglia and the astrocytes that nobody looks at, but that might be there for the Myhre in the Myhre syndrome cases. And if we can develop a technology for the fibrosis or a molecule for the fibrosis, we can imagine to modify that and have a way to get an equivalent for the brain. Not exactly the same, because you are not going to put T cells in your brain, but you can adapt that and make the parallel with the brain and go for the brain there. A lot to do. Very exciting.
A lot to think about, a lot to absorb. Yeah, that's great. Any other questions from anyone? No? We stunned everyone.
Too early for the West Coast. That's too early. Maybe Kate, you could just try to see if there is another day that is easier. Or the other option is to separate the Europe from the US and have maybe an evening in the US and another day for the European, because accommodating from the West Coast of the US up to Europe, it's really difficult to get everyone on the same schedule.
Yeah, I'll ask in the group if there's what would they prefer. Okay.
I don't speak German, I'm sorry. I speak only French and Italian, so doesn't help for the Spanish and the German.
Okay.
Okay.
Good.
If there is no more, then I'll say when is the next one?
It's the week before the conference. So it is on the 20, I want to say 21st. Let me just double check. No, my computer's whirling. 21st, Sunday the 21st, yeah.
21st, okay.
Karin, any questions from you?
No.
That was funny. Nope.
just to know, is it not at all what you are looking for? Am I aiming in the wrong direction when I talk about that, or are you expecting something else? I just want to have you happy and about this kind of meetings. So it's important that you tell me that was not, I'm not interested in that, or
I need to listen the recording with translator to understand. I understand a lot, but not everything. So
okay.
I cannot ask questions.
Okay, okay, I totally understand. Yeah. I think on there might be an automatic translator, no?
Yeah, there is. It's not perfect, especially when you're talking about complex things.
Yeah, I'm sorry, I didn't talk about hearing issues. I see that's we have some patients.
So Kristen, you're talking about Kristen, right?
Yes, I just saw that she signed.
So yes, she can hear. She does have hearing loss. She did have a complete tracheal stenosis, and so she has a tracheostomy as well through a complication of an intubation when she had congestive heart failure. So we learned how to sign after when she was like 14, we started learning how to sign. We did a little bit before that just for fun, but yeah. So, but she had,
Okay.
yeah. So yeah, I just came, Sunday mornings are difficult for us. I just taught Sunday school. We're heading off to church. So yeah, time wise, this isn't the best time for us, but I know we have to accommodate, everybody. So, and as far as questions, I missed most of it. I would love to go back through and listen to it again and see the little bit I heard, I thought was very interesting. But I'm studying to become a nurse. So I've kind of hit some of this, proteins and interacting with things and stuff. So
yeah. So but when I go too fast, just tell me, oh, I didn't understand. Just repeat that, and why is that? And so that we can really so that I can feel where's your level and what you understand, what you don't understand. It's to me, it's helpful to understand what you know, what you don't know, and what you are interested in and not. That's really key to have really nice Q&A and patient drop-in, as Kate called it. It's very important. The interaction is fundamental.
I think from sort of how my brain works as well is that maybe we can do like a summary that is talking at the the visualization or concept of it is you're in the car, it is the brake, it is the accelerator, or it is the book and we're taking out a chapter, or it's in the wrong language, there's a wrong letter, that kind of thing. Just makes it more sticky to a real world, because I'm not a scientist. I think creating something that can just be our reference point that is relevant to a theme and concept in everyday world would help really hammer it home, I think, for everyone when we're thinking about the process, because then we can just start to say, oh yeah, I know that's the foot's on the gas, like it's too much, it's too strong. Like we can just then build up that language amongst ourselves where we're always reminding ourselves, okay,
yeah,
that kind of thing, because it's so complex. we're all marvelous. Like it's just fascinating. Gosh, how complicated we are as human beings.
Yeah, if you think about more than 400,000 proteins that we can create, that's and those are just the proteins, and there are so many other molecules than the proteins in the body. So the complexity is crazy. Okay.
I just
so
did think of something else, but the timeline, I think last time you mentioned a little bit the timeline of like maybe in August, September, we would have maybe the pluripotent cells. Is that right?
Yeah.
Is that the next
Yes.
step? And then after that, what was the next step after that? And the next month calendar plan of that?
Well, we still need to evaluate the different options that we have, but those iPSCs are going to be differentiated into different type of cells. So cardiac cells, neurons, gastrointestinal, bones, fibroblasts, certain type of fibroblasts, because the fibroblasts you have in your ears are not the same as the one you have in your muscles, for example. So different type to reproduce the disease. I'm going to talk to different companies that create models like stenosis models or barrier models, so that they can use the cells from the patient to recreate stenosis model, for example, and we can test molecules or treatment directly on it. So I'm still evaluating all the different options that we have, so that we can choose the best one, the one that is the most relevant for Myhre compared to other diseases. And that takes time because I need to talk to each of them, but that's the priority for Myhre is really to start having those tools. Because there are some groups that are going to have some molecules to test very soon, even now. If we had the tools, we could give them, they would be ready to test molecules now. So we need to hurry up to create the tools to test the molecules.
And the molecules, what are the molecules, the IPCS or the cardiac cells, or?
No, I can't tell you that. That's confidential. Even the team cannot give the structure of the molecule.
I just meant like at what level of are you talking molecules? Are they like cells? Are you what do you mean by molecules?
No, a molecule is not a cell. A molecule is just a molecule is at the very beginning. It's atoms together. So a molecule will be just carbons and hydrogen and nitrogen. It's not a cell. A cell is really macromolecules together, lipids together, proteins together that makes structures like the nucleus, like the membrane of the cell. So the cell is billions and billions of molecules to make a cell.
The molecules that Armelle is talking about in this context that are going to be screened on the iPSCs are potential treatments.
So the iPSCs are the molecules.
No.
The iPSCs are patient-derived cells.
Yeah.
And the molecules are potential treatments that we can see how they interact with the patient's cells.
Okay.
Just to 2 minutes to go back. I'm going to escape, or you are going to be sick, all of you. To go back at that level.
Okay.
So
Water.
you have atoms and for example, water is a molecule. Okay. And then all these molecules, these small molecules, like an amino acid, for example, you see there is a carbon, oxygen, hydrogen, those are molecules. Or here, nitrogen, when you don't have anything, it means it's a carbon. Nitrogen, carbon, nitrogen, hydrogen. This is a molecule. And then you put them together and it becomes a macromolecule. For example, if you have a fatty acid, so you have carbon, hydrogen, then you can have 12 carbon, hydrogen like that. Carbon, hydrogen, carbon, 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 and that's a fatty acid. So it's a long chain of carbon and hydrogen, and at the end you have you can have other type of atoms, and those lipids mixed with some proteins, so an accumulation of a link of those different molecules. You mix lipids and proteins. You put on top of that a bit of sugar, and what do you get? The membrane here of the cell. So here, if you go with a microscope closer and closer and closer to the membrane, you will see that some balls with the lines here, and those are lipids. So inside that, you have molecules that are bound together to form macromolecules that are the lipids, for example, and all the lipids one next to another, that forms the membrane. And you have inside the membrane, you have other small structure, like for example, this one, that's a mitochondria. This is the energy center of the cell. That's where you are going to create all the energy that is necessary for the cell. And there's also a membrane here. So if you go with an electron microscope and you go closer, 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 you will see the molecules that are just aligned like that. And sometimes you will see a big ball like that here. And then again, some what we call double layer of lipids. And that will be what forms the mitochondria with different proteins that are going to help create the energy for the cell. So if you put all the molecules together, they are going to form a kind of structure. It's like this. You have molecules, but you put them together, and at the end you have a structure. So it's the same, and the cell is a big structure with smaller structure inside, so that we call organelles inside, like the nucleus, for example, where you have the DNA molecule inside. So inside the nucleus here, you have all these bones, outside it's sugar, so it's molecules that you put together, bound together, and then it creates a macromolecule, and then you put that in the structure of a cell. You need, guys, just to stop me because I can talk until this evening.
So sorry, so back to the timeline part, you're at the part where you're identifying molecules to have somebody test them. Is that right? Okay. And the molecules specifically are nucleotides or all of them?
No, they are not
might be amino
macromolecules.
acids or fatty acids or
No, they are not the type of molecule that builds the living organism. So they are not proteins, they are not lipids, they are not nucleic acids, they are other type of molecules. A molecule is association of different atoms. So if you associate the atoms in a certain way, you are going to get amino acids that will make proteins, sugar or fatty acids, but you have other type of molecule, like alcohol, water. Those are molecules, but they are not, water is not a protein, it's not a fatty acid, it's not a nucleic acid, it's not DNA, RNA. Water is just water. It's just hydrogen and oxygen and hydrogen together. So you can make some molecules, you just associate the atoms, but it's not because you associate atoms that you are per se getting an amino acid or a fatty acid, or there are many other type of molecules that you can create.
So we're at the step of testing molecules that are nothing like what we talked about today, but I get the concept to see if they can be potential treatments.
Yeah, they are medication. Okay. Like, I don't know, when you take ibuprofen, ibuprofen is a molecule. Aspirin is a molecule, but it's not a protein, it's not an amino acid, it's not a DNA, it's not RNA. It's just a different molecule. It's a chemical compound. So that's in drug discovery, we don't call that molecules, we call that compounds, chemical compounds. So maybe I should use the term that we use in industry, instead of saying small molecules, saying chemical compounds. That's the official term in industry. When I used to work, I would not say to my colleagues, do you have new small molecules? I would say, do you have a new compound?
We're not quite at that step yet, though, where we can test these. We'll be at that step in a couple of months when the iPSCs are ready. We're testing. Someone will test those new compounds on the iPSCs, but the iPSCs are being grown right now.
Yeah, that's the compounds are there, but we don't have the tools to measure them. So we are waiting for the tools.
Any other questions?
All right, so we have the chemical compounds, but we're waiting for the iPSCs, so we can test them on the iPSCs. Okay. Okay.
Yeah, we just started in May, so it takes time to create all the tools, the biology tools, that's the cells, because you need to go to the patient, take the blood, create the cells. That takes time.
And this is not gene editing. This is something different.
It's something different. Yeah.
What is this called?
It's called drug repurposing screening.
Okay.
Who has a cat?
I think Karen's outside. Hmm. Okay, well, that's obviously we've been chatting like an hour and a half. I know that's a lot of information to take on board. And perhaps at the beginning of each session, we will just have like a one single slide that's just like a recap for everybody on just the very basics of like what we're talking about in like layman's terms. And then, you know, we can move on from there and then always revert back to remind ourselves, okay, yes, this is the process we're talking about. So yeah.
Kate, are you going to create these slides?
I'll create that. Armel and I are going to be the perfect kind of meeting of the technical and Armel, and I'll try and make it into some traffic light system or something. So that
translate, yeah.
Yeah, I'll translate your stuff.
Where do we find the videos?
They're just on our YouTube channel. So we have a YouTube channel. You can sign up and it will alert you to new content. And you can also link off our video page on our website to find it. I'll send the recording note after the group to everybody who signs up. And I'll put it in the newsletter as well. If you've not signed up to the newsletter, please do so. You can sign up at the bottom of our homepage.